Good morning, everyone. I'm Simrinda Costa, and on behalf of Altum Technologies, I'd like to welcome you all for another very informative virtual session. So, our session for today is entitled Plant Secondary Metabolites as Effective Drugs in Modern Medicine. Now, before we begin, a little bit about plants used in medicine. So, during this entire series, we've seen that in addition to being used as a source of food, plants also have a very vital use in the field of medicine. And we see plethora of scientific studies on plants that have revealed the presence of innumerable types of medicinal compounds. So, in the early years, the nitrogen bearing alkaloids were thought to be the only active principles responsible for the physiological properties of plants, but later on, detailed investigations showed that other groups of compounds, like steroids, phenolics, etc., had a very significant biological activity when studied in both in vitro and in vivo models. And in the old medical books, like the British Pharmacopoeia, most of the pages have been dominated by plant drugs, but in due course of time, we see that they were slowly replaced by more effective but highly side effect causing synthetic drugs like antibiotics, sulfa drugs, etc. But in spite of all this, plant based natural drugs are still used in treating many chronic and baffling diseases because they are pretty effective and they tend to cause the least amount of side effects. So the age-old traditional medicinal systems like Ayurveda, Unani, Siddha, which mostly use plant drugs, come in handy for choosing the right plants for research to isolate more and more new drug entities in treating diseases. And with the advancements in technology, we've seen several sophisticated instruments that are used in the present day research, wherein even milligram amounts of phytochemicals are studied for their detailed chemistry and biology. So these studies give us ample of scopes for the discovery of new drugs. So let's understand the perspectives of medicinal plants used in indigenous medical systems that have several interesting pharmacological properties from our speaker of the day. So it gives me immense pleasure to introduce Dr. Sukhu Kumar. Dr. Sukhu Kumar was the Dean of Research in Savita Deemed University, Chennai, and is currently a scientist in the Division of Endopharmacology and Phytochemistry at Loyola College, Chennai. He's a visiting professor in the University of Canada and VP Pro Biotech Pondicherry as an expert member of Institutional Ethics Committee of Sri Venkateshwara Medical College, Pondicherry, and Research Advisor Council of Meenakshi Deemed University, Chennai, where he was sharing his experience and expertise for the benefit of students. Prior to these, he was an associate professor at the University of Technology and Applied Sciences at Muscat, Oman, and served in the Ministry of Ayush as a scientist. He was also a visiting faculty in two colleges in Chennai and worked as a research officer in Tamil Nadu Medicinal Plant Farms and Herbal Medicine Corporation on deputation. He was nominated to various government boards, professional bodies, and institutional ethics committee, where he played a very significant role. The National Assessment and Accreditation Council have appointed him as an assessor. Dr. Sukhu Kumar holds two master's degree and has obtained a PhD for his research on the studies on the chemistry and biological activities of plant products from the University of Madras. He has had a postdoctoral stint at the University of Virginia, USA, and during his professional career spanning more than 40 years, he did noteworthy research on traditional medicinal products, indigenous drugs, formulations, metallic drugs, active phytochemicals, chemotaxonomy, field botany, toxicology, environmental pollution, bioinformatics, and traditional foods, and he has published over 90 research papers in leading index journals. He has also edited four books and wrote eight book chapters, besides presenting 60 papers in conferences, seminars, symposia. He has bagged the Best Paper Award twice, one at Indian Pharmaceutical Congress held at Hyderabad and another at National Conference on Indian Association of Biomedical Scientists in Chennai. He has delivered 65 invited lectures and chaired sessions in many national and international conferences, as well as acted as a resource person in workshops. He has guided 30 scholars in their PhD, MPhil, MPharm, and MSc programs, and held editorial assignments such as associate editor, member of advisory committee, editorial board, and peer reviewer of reputed journals. Notable mention of his expertise is cited in the International Directory of Experts in Medicinal Plant Research, published by the University of Massachusetts, USA, and Who is in Science and Engineering by Marcus, INC, USA. Now, without taking much time, I'd like to hand over the platform to our expert. So, welcome, sir, and I invite you to take over the session.
Oh, thank you very much, Ms. Simran, for detailed introduction. And already you briefed what I'm going to talk in this session. At the outset, I would like to thank uh, Altem Technologies for arranging this presentation. I'm very happy to share some of our experiences with the young minds so that it will be useful for them in their research and in their degree programs. As told, the audience for this program consists of baccalaureate, masters, and business scholars besides industrial research people. So within this 30 or 40 minutes, I cannot do justice for all. So I'll be starting with the very preliminary uh, details, and I'll go further and then explain what all things we have done. And especially my interest is to focus on the use of traditional medicines. We have rich culture of traditional medicines, how they're useful in getting effective drugs to be used in the modern medicine. So this is very exciting area going on in a lot of places around the world worth of billions of dollars. So my topic I've chosen is plant secondary metabolites as effective drugs in modern medicine. Now the next slide please. Next slide please. Yeah. In this presentation, I'll be covering the importance of plants, then research on plants, secondary metabolites and classification, select my secondary metabolites and their activities, and our own work on secondary metabolites, and what are the challenges we face in the plant research, then alternate methods for isolation of production of secondary metabolites, and how nowadays modern fields like biotechnology, genetic engineering, bioinformatics come into play. Then I will be concluding with a summary of what we have done so far, and then change with acknowledgement. The first slide shows, maybe the next slide please. The next slide shows, hello, next slide please. Yeah, there are some interesting facts about plant research. So if you understand this, you can appreciate the presentation well. Okay, in the early stages, more botanists were involved in this field. So once upon a time, sometimes in 1910, 1920, and all that, if you go through research papers and books, only mostly botanists only were involved in the plant research. They identify a lot of plants, and then preliminary studies they do and find out what are the types of components present. That's how they stopped there. Then came the chemists. Chemists are very curious to go deep. Even in the atom, they go to center of the atom, nucleus, and electron, proton, neutron, like that. So chemist's mind is always very deep. It goes deep, and then they started looking into the compounds, the types of compounds present, and the process they isolated many new compounds. And then to pharmacologists and microbiologists, so what they did, they started analyzing these things to study the activities. So it has taken, the plant research has taken, went to the next step. So further, no, afterwards, in the 60s and 70s, some pharmaceutical scientists entered the field and they started their own field. They named it as a pharmacognosy. So what they did, they identified the plant, they found out the components, and then they collaborated with chemists and botanists, and then they came out with a lot of compounds, and then they made use of these compounds to convert them into pharmaceutical entities. Then now, after the advent of biotechnology, field of biotechnology 2 decades back, so now more biotechnologies are coming inside because there are lots of issues in using the plants, particularly in bulk. Suppose if you go on destroying the plant, definitely it will affect the environment and ecology. So now biotechnologists, what they do, they find similar methods for the extraction of these secondary metabolites. And next, we do not know which field is going to come inside. But I'm sure many people are coming inside. So now, at every stage, the research has got enriched with more inputs from the fields in the people of above fields. So now, the plant research has become a multidisciplinary science with openings for many researchers worldwide. So now we cannot say the plant research is confined to only one field. Now it has become a broad field. See, the college where I am now going as a visiting professor, now more research is going in the zoology department on the medicinal plants because they want to identify some of the ingredients, some of the active principles of secondary metabolites for some of the zoological issues, some of the animal related issues. Okay, next slide, please. So we all know plants. Why? Next slide, please. So plants, ever since man came into the universe, they started using the natural materials, more of plants. So we are all plant consumers. In one form or other, the plant is being used. May the next slide, please. Yeah. So plants are used as a food. We all know we are taking vegetables, leaves, green leaves, and many things. Our food is mainly from the plant source. Then clothing. So nowadays we use synthetic clothing, but once upon a time, even presently, we go for cotton cloths. The cotton plant. From the cotton plant, we are getting the, cotton and then making it thread and using as a cloth. So shelter, we use timbers from the trees for shelters. Now we are going for environmental friendly houses. So now, because people do not want more air conditioners because they affect the environment. Because the greenhouse gases, more greenhouse gases are released. So they want to reduce the use of electricity. So they want to construct houses or some industries. Even in some of the industries abroad, they are constructed environmentally friendly materials. So in that, the trees, plants play vital role. And of course, medicine. You know very well, the age-old medicine, our grandma medicine, which in Tamil we call Pati Vaidya. It means our grandma medicine. So supposing for child, some children fall ill at home. In those days, I don't know now present day grandmas give such medicine. In our days, so our grandmas used to give the extracts or the decoction made of some plants to cure the small diseases like cough, cold, fever, body pain, those things. Okay, so medicine, the plants and medicine are both are highly close, and this has started ever since the man has come to the field, come to the world. See, when man was going for, just imagine the first man born in the world. So definitely he was searching for food for the survival. So he got only from the plants. When he got injured, or when he got affected by the animals, so he got wounded by animals, he has to seek remedy only from the plant. So like that only the plant science has developed. Next slide, please. So as food, how plants are useful. Next slide, please. Hello. Yeah. As food, plants contain essential nutrients, fibers, vitamins. See, nowadays, lots of diseases are due to food. Because modern food, the colors added in the food, the taste enhancer, the other chemicals, preservatives, they cause a lot of health problems. So what people told, doctors tell, use more of fiber food, okay, and which contains vitamins or the carbohydrates, proteins, all which is kind of essential nutrients like zinc, copper. These things are present in the plants. So the plants and food are closely, they're both embedded. Next one. Here's the next slide. Yeah. So the plants contain majorly the three types of components, namely primary metabolites, secondary metabolites, and cementite. So from here we start our discussion. So primary metabolites, what are the primary metabolites? What are the secondary metabolites and cementite? Next slide, please. We will discuss the next slide. The primary metabolites are present in plant in large amounts. See, for example, starch. Supposing in the rice food, we have more of starch. Then ethanol, in the sugar cane, we have ethanol. Lactic acid is present in the milk. Okay, some amino acids. The citric acid present in the lemon. So these are all large amounts are called primary metabolites. They are the central metabolites. Okay. Secondary metabolites are present in very small amounts. They are not directly involved in the plant metabolism like growth or development or reproduction, but as important ecological functions. Typically, they are present in the present in the restricted organisms. They are not present everywhere in all parts of the plant because plants contain a lot of anatomical parts: roots, bark, leaf, flower, stem, like that. A lot of components, a lot of anatomical parts are there. So these secondary metabolites are confined only to some of the anatomical parts of the plant. And the third one, cementitides, or information carrying molecules. Just like we communicate, plants also communicate. For that, they use cementitides. They are further classified as primary, secondary, and tertiary cementitides. So these genes, mRNA, polypeptides, are group of cementide. Now our focus is on the secondary metabolites, or we can call simply phytochemicals or natural products. Next slide, please. Yeah. So the plant secondary metabolites are broadly classified into many groups. Okay, based on the types of atoms, types of groups present in it. Here, for example, alkaloids. So alkaloids are nitrogen-bearing compounds. It doesn't mean that all nitrogen-bearing compounds are alkaloids. Okay, there are amines also. Okay, so they are not alkaloids. So specifically, the organic compounds containing nitrogen are alkaloids. Then steroids. The plant steroids are very good for health. Okay, then terpenoids. Phenolics. Phenolics are a broad group of classification. It consists of flavonoids, coumarins, like that. Then anthocyanins. So these are some of the classes of the plant compounds or the secondary metabolites present in the plant. What they do in the plant? So this question I always had when I started my research some 40 years back. I used to ask my colleagues, senior colleagues, and my teachers, my guide, and all that, what these compounds do in the plant. That one only understood. They give taste. Okay, they give color, smell to the plant. Very well. Supposing if you take coffee, it has some taste. You take tea, it has different taste because coffee contains caffeine, tea contains theobromine, theophylline. So these two different phytochemicals. Okay, second metabolites are there. And they act as safeguards against animals. When the plants grow, naturally they are attacked by animals because plants are food for animals and humans. So some of the plants we ourselves do not touch, say telling that they are toxic. And some of the plants animals do not touch because they are toxic to them, they avoid. So for that, there are some specific secondary metabolites. As well as insects. Insects are very common. Even if you store half a kilo rava in our, in our house, some insects come. Or the maida, atta. Everywhere, insect attack is very common everywhere. So these plants, create their own defense system through these plant secondary metabolites. And not only that, these secondary metabolites are intermediate or the byproducts of the plant metabolism. Just like we have metabolism, when we take food, what happens? The food from the food, nutrients are absorbed by the body, and waste products are excreted. Like that, this kind of cell system is there in the plants also. So these secondary metabolites play an important role as immediate, intermediate, or byproducts of the plant metabolism. Next slide, please. Okay. What are these utility of these plant secondary metabolites? They are substrate for biochemical reactions. Most of the biochemical reactions, they are substrate. They are cofactors of enzymatic reactions. They are inhibitors of enzymatic reactions. Absorbents which bind and eliminate undesirable constituents in the intestine. This is very important factor because most of the people suffer from constipation because they don't take adequate amount of vegetables or natural food. So these the secondary metabolites present in them actually bind to these undesirable elements in the body and then they eliminate. Scavengers of the reactive and toxic chemicals, in particular free radicals. So whenever we take unwanted food, the food which are not suitable to us, or the medicine which are not suitable to our body, so they generate a lot of free radicals. These kinds of plant secondary metabolites, they act as scavengers. They absorb them and make them inactive. So they enhance absorption and stability of essential nutrients. So when you take the nutrient, it won't be absorbed just like that. It needs some catalyst. So these kinds of plant secondary metabolites act as catalyst for that. So they have a selective growth factor for benefit of bacteria. So in our body, billions of bacteria are present there, so, both good and bad. So particularly, for some kind of beneficial bacteria, these secondary metabolites use as a growth factor. Then fermentation of substrate for benefit of bacteria. The fermentation is a process which is happening in the body as well as outside. So particular food industry and medical industry, medicine industry, they use it. So this is used as a substrate. And selective inhibitors of release these intestinal bacteria. Some of the bacteria, unwanted bacteria, they are killed by the secondary metabolite. So the secondary metabolites play vital role in a human system as well as in animal body as well as the plants. Next one, please. So what these secondary metabolites do in the living organisms, particularly humans or in animals? Okay, they suppress the growth of unwanted tumor cells. Particularly, these type of compounds are known as tocotriols or uh, tocopherols. Then carotenoids, they are antioxidants. They protect against a variety of tumors. You know, the tumors nowadays, cancer has become a very common disease. So every 10th person is getting cancer nowadays. Why? Because of our lifestyle, because of bad lifestyle and dangerous food habits. So they create, okay, particularly a lot of harmful chemical-based foods. They create this kind of tumors. So these kind of natural foods containing these secondary metabolites, particularly carotenoids or flavonoids, those type of things, phytosterols, phenolics, flavonoids, catechins, they have lots of beneficial effect over living organisms. Next slide, please. Likewise, the flavonoids. Flavonoids are very important phenolic compounds. Next slide, please. Maybe the next slide. Yeah. The isoflavonoids. The these are isomers of the flavonoids. They are used in cancer. Osteoporosis treatments. Osteoporosis is a very common disease, particularly women. After 50 years, they get joint pain, hip pain, neck pain. And osteoporosis is one of the serious issues in women. For them, the isoflavonoids present in the plants. They are the secondary metabolites. They are very helpful. Anthocyanins. They actually stop this mutagen. When cells mutate, then only these kinds of problems come. The cell proliferation takes place and cancer occurs. So this kind of anthocyanins, they help. Then glucosinolates. So they activate the liver detoxifying enzymes. The liver problem is now very common. Once people tell, once people are told, only people who take alcohol or drink unwanted in the drinks, synthetic drinks, they only get liver problems. Nowadays, non-alcoholic steatohepatitis, some of the diseases come. Even people who do not consume alcohol, they get other liver problems. So these kind of compounds present in the plants, they actually help to detoxify the enzymes and carcinogen inhibitors. Then, like the indoles, saponins, phytoestrogens. These estrogens are very, very important, especially for women, because once they cease to, when, once they attain the menopause stage, actually the estrogen production stops. In the case, they have to take more of this kind of plant products, particularly which are rich in secondary metabolites, so that to reduce the menopause symptoms and increase the bone growth and other things. My last PhD student who presented her defended her thesis last November, she worked on the postmenopausal problems of the women, particularly nowadays more women in the premenopausal stage itself, they get the symptoms of cardiac diseases, and in the perimenopausal stage, we could diagnose them. So my last PhD student, my 12th PhD scholar, she worked on that one. We got 4 papers published, and the thesis was very much appreciated. In that, we dealt with this kind of estrogenic treatment. So instead of going for modern tablets, we can use the vegetable products which are rich in the secondary metabolites. Then lignans. So these are all present. These are all present in the very very rare kind of plants which reduces cancer and heart diseases. These kinds of diseases are very common. So the use of secondary metabolites are very much useful for nowadays the present day life. Next one. Next slide, please. Yeah. So particularly the secondary metabolites nowadays, they are targeted. They are targeted to treat some of the different typical types of cancers, which are very common. You know, they protect the plants and some of the excellent anti-cancer properties. For example, winged houseflies. In some states, it's called Ethirajan. It is available just like a weed. It's available everywhere in our country. So from there, winter alkaloids, wind blasting, they are now used, particularly for childhood leukemia. These two products are used as not as a product as they are used as a derivative in order to enhance the availability, bioavailability, and other things. Then camptothecin, because this is very good. This also has been shown to have very good anti-cancer agents by National Cancer Institute, USA. Then paclitaxel, taxol from Taxus brevifolia. We will discuss about it later. Then portfolio toxin from Porphyrophilodendron. It's also one of the important anti-cancer compounds discovered in 70s and 60s and 70s. Okay, next one please. So these anti-cancer compounds actually how they work, how they attack the cancer cells. So these are some other example. Say for example, Chinese medicinal plants, Piscolis indica. This plant is also available plenty in our country, particularly as an ornamental plant. Even in now to speak, we'll have this one. So this one, has been identified after screening 253 extract of the plant, 75 plants with the research group. They collected 76 plants which are used in traditional Chinese medicine. I mentioned as cesium. In fact, now they taken 253 extracts and they identified this particular compound cucurbitacin F. So it has significant growth inhibitory activity against leukemia cells. Okay, this has been observed in 23 extracts out of 80 traditional medicine plants, traditional Chinese medicinal plants. So these are the such of the compound. Next one, please. So these are some of the examples to show how secondary metabolites play a vital role in our day-to-day life as well as in medicine. These are already being used. Next one, please. Yeah. Then one is Milteiron. So this isolated another Chinese medicinal plant, Salvia vitorosa. So it has very good. It has been studied by National Cancer Institute cell lines, and it has got very good anti-cancer activity, particularly sensitivity against the cell lines. Many cell lines are sensitive to this one. A lot of work has been done, and the papers have been published. Next one, please. Yeah, flavonoids, as I told you, flavonoids and leaf edema. Leaf edema is some of the very common thing in the among the people, which goes mostly unnoticed. So this actually, uh, has been cured or been controlled by managed by flavonoids present in the plant, which are these flavonoids are present in the fruits and vegetables, and they have interesting structural features and have many biological activities. In particular, in the leaf edema, major action is by a particular mechanism on the microcirculation of decreasing number of micromolecules leaking from the blood vessels is one of the important mechanistic pathway in the lymphedema process. Okay, in the disease onset. So this particular flavonoids, group of flavonoids we have studied, and it is really very good research, and people even are treated in some parts of the world. Next one, please. May I have the next slide? Yeah. Coumarins. See, nowadays, most of the researchers are going for small molecules because big molecules, they have their own problems. They can't cross the blood barrier. So blood barrier crossing is one of the important factors in the drug research. So the drug has to cross the blood barrier so that it can give a beneficial effect. So these coumarin type of compounds, they bind to plasma protein and they arrest some of the leaky microvessels, and in the process, they reduce the impact of lymphatic responding activities in the body. So the present drugs for the lymphedema, they have hepatotoxicity. This is another problem in drugs. So whenever a good drug is recommended, it has some side effect. A good drug to treat the disease and control the disease, or even completely destroy the causating organism, causative factors, but they have side effect. Particularly this type of coumarins, they have hepatotoxicity most of the time. Whereas these coumarins, particularly some derivatives, they do not have this one. They are very liver friendly, and they go along with the liver function and cure the lymphedema type of cancers. Next one. Next slide. Yeah. So many people ask, even this question, same question I asked when I started my research. Why you have to isolate more compounds? Why you have to search more plants? Why you have to kill more plants and to isolate more compounds? Because nowadays in agriculture, new types of insects are generated. They are in the field. Then rodenticides, pesticides, herbicides. In agriculture, we need different types of agents to control these things. Otherwise, we cannot get food. The food loss is increasing everywhere. Nowadays, by modern methods, they are controlling. Okay, because whenever the foods are grown, food plants are grown, the insects, rodents, these particular pesticides and herbs, the weeds, they affect the growth. And because of that, yield decrease. So when the yields are going down, the farmers do not go for cultivation. So when the cultivation goes, the food reserve goes down. So when food, there's a starvation. It's a demand for food. So starvation. Many things are there. It has impact on the economy, health of the people. The country face a lot of problems, and even people go for, internal conflicts may arise. So naturally, for this to control this, Naturally, we need more plant research. Then medicine, as I told you, new diseases we have seen, COVID-19, no medicine at all. So people are searching frantically what to eat, what to take to prevent death. So now we need new drug entities and for the same diseases. And of course, now the researchers are warning, the medical researchers are warning that in future we are going to get more diseases because of the climate change, because of the lifestyle modification, because of the pollution issues. So then food materials. So nowadays we have to preserve. So we cannot go for preservatives or coloring agents or sweeteners or taste enhancers which do harm. So we need natural materials. So the plant secondary metabolites are very suitable for this. Next one, please. Why plants? As I told you, see why you need plants? Because plants contain innumerable number of compounds with wide spectrum of chemical structures. As I told you in the beginning itself, the compound with different types of compounds we have seen, alkaloids, terpenoids, sulfonides, steroids, like that, there are innumerable number of classification of compounds. Another advantage is, as of now, we know only 1.2 lakhs of the taxa. Plants only we know. And how many, only less than 10% has been scientifically screened. So it will still more scope for finding more materials, more new types of compounds, and then we take drugs also. So in addition to that, India, countries like India, we are very fortunate to have traditional system medicine. As I told you in the beginning itself, my focus will be more on the traditional system medicine because they are virtual gold mine for researchers. They have lots of material, lots of information given by our ancestors. So if you go deep into the research, we can get lots of information, lots of plants and other materials for research, so that lot of new drugs can be invented in future, put to use. Next one, please. Now let us go and discuss about the traditional medicines. We'll go a bit away and then we'll come back afterwards. See, for example, because some people may not be aware, what are the traditional medical systems? So our country is very rich in heritage and civilization and culture. And these things were founded several hundred years ago. So we know about the religion here. We know about Vanasastra. Once Indians were expert in 64 arts. We were expert in astronomy, astrology, mathematics. The zero was discovered by us. And the modern mathematics, Ramanuja, we gave mathematician to the world. Ramanuja, like that, Bhatta. Now we do a lot of space research. Those things have been done already. So we have Ashtanga Hridaya and what butter. Many things are there which which we have to revisit and then find out what are the important missing. Now whole world is looking at India to go into these things and find out. These systems grew along with the culture and tradition of the country and the race, and it faced lots of onslaughts of the external forces. India never invaded any country, whereas many countries invaded us or nearly for 350 years, if you see the history. So they spoiled, they actually destroyed our traditional things. They destroyed many worship places, they destroyed our culture, the medical systems, many things. And because of that, the gap, other systems, the European system came, and we are very comfortable with that, without minding the side effects and other harmful factors. Okay. So still these traditional systems, serve considerable section of the society in many parts of the world. So when I say this, we have to go for traditional medical systems, and we are not against modern medicine. So okay, modern medicine is a must for the present day life. We need it for emergency, for diagnosis, and those things. Whereas for prevention, the traditional medical systems are very good because the modern medicine system never advocates anything for prevention. They only cure. They only go for treatment to cure, attempt to cure. Okay. Whereas or manage the disease. Whereas the traditional medical system, if we incorporate in our life as a day-to-day affair, definitely we can be safe. It is a precautionary. It can be a prophylactic thing to prevent us from the diseases, to increase the immunity. So you were saying during the COVID, what they told, those who were weak in the immune system only got affected. So once many people whose immune system were strong, they escaped. So we see many people who succumb to the disease had these problems. So these some of this, see, a lot of every country, not only India, like that, even Indonesia, they have Balinese medicine. In China, Chinese medicine, Egyptian medicine. Like that, we have in Africa, we have Zulu medicine. Like the many systems of medicines are there, but in due course of time, due to lack of interest in these things, many things got perished. Whereas Indian system of medicine now has seen a lot of, renovation. We can say renovation, or we can facelift. Now lots of new things are coming up. New institutions, more graduate courses, universities are coming up, and a lot of foreigners come and they learn these things. So once it was like that. So many people abroad, they came here. Even Chinese travelers, they are recorded. The people from China came and learned this system medicine and went back to the country and spread those things. Because in those days, no demarcation of countries like this as we have today, like state, district, country like that, a continent like that. So people are freely roaming from one end to other. So they brought their culture here, they shared, and as well as the old people went there and shared. Like that only it has spread. This system spread all over the world. So some of these medical systems are recognized by WHO and included in the healthcare programs of many countries. So during COVID-19, as I told you, we have seen most of these medicines used all over the world to save the people from death. Next slide, please. Yeah, as I told you, one disease of medicine broadly consists of Ayurveda, Siddha, and Unani. You know what's Ayurveda? It actually, uh, it was actually the literature is in Sanskrit language. It originates from North India. Siddha, the literature is Tamil language. It originates from South India, particularly southern parts of India. Whereas Unani is a Persian medicine which traveled along with the Mughal travelers as well as Mughal invaders came to India and got naturalized here. And the great philosophers, the Greek philosophers, so basically Greek medicine, the Greek philosophers, they contributed a lot for the Unani medicine. It has undergone a lot of changes. Now it is very much in vogue in India. Government of India is recognized as one of the Ayush systems. Ayush, Ayurveda, Unani, Ayurveda, Yoga, Unani, Siddha, and Homeopathy. These five things, now Government of India is constituting. A separate ministry is there, separate ministry is there. So when we were working in this ministry, it was the Department of Indian System of Medicine at the time in early, 80s and 90s up to 2000. These things were under Ministry of Health and Family Welfare of Government of India. Now separate ministry has been created. Next slide, please. So what are the importance? What are the specializations in this field? You see, in addition to that, we have a lot of folklore medical practices. They are confined to some native groups of tribals, like tribals. For example, Goans of Madhya Pradesh, the Thoras of Nilgiris, the Malayalis of Police, the Kani tribals of Kerala. They have their own traditional systems. Unfortunately, these things do not have any written documents because those people are not, in our ancestors, in general, our ancestors, most of the people know at the time no education, no written language. So only by word of mouth they were passed down from one generation to another. So one link disconnects, the whole thing vanished. So like that will it happened. But luckily, when education came, when the people started writing on the palm leaves, these things were recorded. Some of the things. So selected libraries, particularly in Tamil Nadu, Saraswati Mughal Library in Tanjavur Palace, and Indian Institute of History of Medicine in Hyderabad, and many institutions, even I think National Museum in Delhi and Calcutta, they have these palm leaves here. And you have to decode because they would have written it in their own way. So now we have to decode it. Then we can make use of this information for our research. Next one, please. Next slide. Yeah. So the drugs. So these traditional system medicines, they use plants, metals, minerals, animal products as drugs because they were the only thing available in their vicinity. So they were used either singly or in combination. The combinations are formulations. Various formulations. If you go to Ayurvedic doctor, he will give churna, legia, grita, thaila like that. Prepared by these plants or metals or minerals. And they used to prepare the conventional methods. They use stone grinding machines and heating. They use firewoods and then open mud parts. With that only they process those things. So some drugs were processed in sunlight. So now we call as insolation. Okay, it's called Suryaputa. So now only we start using the sun's energy for our electricity production and those things. Whereas our ancestors started using those things. So the exact science behind this process are yet to be explored. Why I'm including these things? Because these things need to be detailedly discussed and it gives scope for many scientists for another 200, 300 years. Next one, please. Next slide. Yeah, these are some of the active secondary metabolites from plants. So these secondary metabolites, the active compounds, actually were recorded in the as given by Ms. Simran in the introduction. So these things were found actually placed in the old pharmacopoeia, like British pharmacopoeia. These were the drugs ones. For example, Overface serpentina. The whole plant was used in Indian medicine as anti-hypertensive. Nowadays more people have this kind of hypertension, blood pressure. So in those days also people had only select minimum number of people had. So what happened, these plants they've been using, but scientists isolated the responsible compound reserpine. Then reserpine became a modern medicine. Then cinchona. So people were suffering from malaria. So from malaria plants, Cinchona, actually we had one, the Cinchona Corporation in Tamil Nadu government. They closed it some 20 years back in Ooty, in Uttarakhand district. So they had Cinchona Corporation. And they were growing the Cinchona plant. From the bark, they started actually isolated quinine and alkaloid, which was very effective. So why they closed it, that one? Because, mosquitoes started developing resistance against quinine. This is another thing, very, very bad thing is whenever we use some new drug, the causative organisms they develop resistance. So naturally we have to go for a derivative or a new drug. So that's why more drugs are in use nowadays. Okay, then Papa somniferum, analgesic. It's actually it's morphine. So Papa somniferum we are using in our kitchen. Actually, it's analgesic. Particularly, I remember when in those 40, 50 years back, when our children, they cry due to pain and other things. Mother used to give the seeds, they dissolve in water and give the water decoction, or not decoction, actually cold extract of that one to the child, it sleeps. So the pain, everything disappears. So later on, the scientists found out the morphine alkaloid is responsible. So morphine is used even today as morphine sulfate. And modern surgeries, they give morphine. Okay. Then Sorelia califolia. This is one of the important drugs used as a lecoderma, white patches in the skin. So Soranin is the active principle. Even today they are used. The Soranin is being used in the modern medicine. Then Berberis aristata. Okay, this anti-cholera. Of course, now cholera, the incidence of cholera is very less. The berberine, the alkaloid berberine is the responsible compound. Then coffee arabica, we know very well. We all take coffee regularly. Whenever we feel tired, so immediately we take. It's a central nervous stimulant. Supposing if I work in the lab between morning 8 to 11, I get depressed. So immediately I go to canteen, take a cup of coffee, so I get energized. It's only short, it's short stimulant. It should not lead to addiction. Okay, the caffeine is the active principle in that one. So mecabomuneri, okay, that's it's a brain tonic. So Percocets A and B is being marketed in India by some pharmaceutical companies. So like that, we have lots of materials from the plant, particularly the secondary metabolites they are used in the market, and not in the same form. They, due to marketing and other issues or approval of the authority issues or side effects, some of these drugs are changed and they made as derivatives or combined with other drugs and made as a new product and patented and marketed. Next slide, please. Maybe the next slide. Yeah. Some of our work on plant secondary metabolites will go fast. This is one of the plants. Actually, we worked on many plants. I want to highlight within this short time. I can't project all these things. Select things I want to project. And these are the plants of Pistimera grahami, which we collected from Pune, from the Western Ghats. 100 kilometers in the Western Ghats from Pune. We went there and collected near close to Bhima Shankar hill, Bhima Shankar temple. It's one of the 12 Jyotirlingas temple there, Bhima Shankar hill. It's called Bhima. There we collected this plant. Next slide. We got nearly 17 terpenoids. We isolated from this. It formed a part of my thesis work in 19, for which I was awarded PhD by Madras University in 1992. I worked for nearly 12 years for my PhD work because I worked on lots of plants and of course I was traveling and the things it took a lot of time. It took more time to work. So these are four important trited nuts which give very good name to our work, our group, and we publish these papers in international journals, published from USA, UK, and other places. So these are all very important, very exciting structures. So it took a lot of time for us to integrate the structure. It was very challenging and very interesting also. Next one, please. So another plant we worked on was Ceropigia juncea. So it's actually called, in Ayurveda there is one a drink called soma. So this is considered, this plant is mentioned as one of the source goods of soma. So we took interest in that one. We collected this plant and very succulent plant. It means contain more of water, 90% water. So I collected 28 kgs of this plant, traveling to different places for nearly 6 months, and then on drying I got 2 kg. So for 1 kg only dry plant, from that I isolated 1 gram of a new type of compound. Actually, we started a new series of compound called the Ethirajan. Next slide, please. So we emulated the structure. It took nearly 2.5 years for us, and we interacted with the people in the University of Glasgow, UK, University of Calcutta, National Cancer Institute. Every lot of places we went there and got this benefit. Next one, please. This is The structure of the compound is given here, serpentine. With lot of time, IUPAC name is very big. From the whole plant we got it. And the isolation, uh, the paper we published in Phytochemistry 1991 has received so far 92 citations. Whenever you publish a good paper, it should receive citation. Many people should mention. So soon, I think by this my ARI it will hit century. So a lot of our papers crossed the 300 citations and all that. Then anonymously people took study on the exocrystallographic studies to study the the structural pattern, three-dimensional structural pattern, and they published in Abstract Crystallographica, this one. And then biological studies we did, and then that also earned a lot of good name for us. Next one, please. So just I'm passing through. We have done a lot of work within this specific time. I can't give more details about this thing. Next one. If you are interested, you can contact me later. We will discuss. So these another plant which we collected when we went for plant collection near Kodikari Forest in South Tamil Nadu. So we got on. We got it near the seashore. So it is not a medicinal plant actually. It's a lichen. Lichens are symbionts of organic fungi. So this is a new type of plant. It's one very close to the littoral forests in the forest adjoining the seashore. It grows very well. Next one, please. So from that, we isolated a very good compound like usnic acid. Usnic acid. This has been already reported from other lichens also. The first time we reported from this particular lichen, Rapsilla montagni, and we studied the anti-inflammatory activity in acute and chronic model. It formed part of one of my the M.Pharm students, and we published the paper in Phytotherapia. The impact factor, present impact factor is more than 4 point something. Then so far it has received 209 citations. It means lots of work is being done on this particular plant, this particular secondary metabolite. So these are the encouraging things for us to look into more plants, to more types of secondary metabolites, and we are still working on it. Even though I retired from service, I am collaborating with a lot of people abroad and India and encouraging the youngsters to take up such kind of work and continue the research. Next one, please. Maybe the next slide. Yeah, this another plant, one of my PhD students worked, and is Trivia polycarpa. So from this we isolated one compound, macronic acid, actually, which has been found to have effective in HIV, inhibition of HIV activity. Because in those days when this paper was published, the HIV research was very, predominant. Most of the people were studying the compound. At the time we came across this one, but we also did not do the HIV studies, but we isolated the compound first time from the Indian medicinal plant. Next slide, please. Madam, I have the next slide. Yeah. This macronic acid isolated from the roots of the plant Trilia polycarpa, first time from the Indian plant. So this plant was this particular compound was earlier isolated from an African medicinal plant called Macronia afranica, and Africana, and it was found to have HIV one reverse transcriptase inhibiting activity. It's one of the important mechanisms in stopping the spread of the HIV. It formed a very good particular compound worked very well. So first time we isolate this compound from the Indian plant. Next one, please. Yeah, this work has given a lot of encouraging results, and two of my PhD students worked on this, and I'm still working on this one. Next one, please. So this is the Emblia ribis, and it's an Ayurvedic plant, which in Ayurveda is called Vidam. In Tamil, it's called Vayu Vidangam. In Siddha medicine, in Ayurveda, it's used as Vidam, particularly for to eliminate the worms from children, particularly from the infants. They give this particular powder of these fruits, small amount, so that the worms are excreted from the body. Okay, because worm problem is a very serious issue in the infants. So this particular thing is used in the Ayurvedic medicine, Siddha medicine. We took up the plant, and next slide, please. And we isolated a compound called embelline. It's one of the very interesting quinone compound, benzoquinone compound. The compound is already known. They isolated and they studied only two activities. They studied anti-emetic, the killing the worms in the stomach. Then anti-fertility activity we studied. We took up my first PhD student in '92. She took up the work and she worked on anti-tumor, particularly fibrosarcoma. The fibrosarcoma is a oral cancer, particularly people who smoke or tobacco use, users, they get affected by this. We studied these four important properties: anti-tumor, antioxidant, antibacterial, on the effect on lipids and glycoproteins. We published some papers. So from my thesis, I published some papers, and my first student also published some papers. The first paper in chemotherapy. It's out and out a clinical journal published from Switzerland. So far till date it has got 241 citations till date. So this particular work is given is very good name. You go back. Can you go back, madam? Yeah. So then next one, we got another paper where we two papers in phytotherapy and medical science research. And this particular activity has been uh, further confirmed by Jagiellonian University in Poland afterwards. So that's not visible in the slide. I written in the footnote. I think can you reduce the slide size and yeah. Okay. So, Emily, that we didn't stop. Recently, a couple of years back, we took the work for anti-Parkinson disease in model, in a nice model. Parkinson disease also increasing in the country. The last week also I read a report. Lot of people, particularly after 6 years, they are affected by that. It affects the brain. Okay, this is due to many factors, for smoking, drinking, or many causative reasons. So we found that it's a potential drug for Parkinson's disease treatment in the mouse model. So in combination with the L-DOPA now, as of now, there's no treatment for Parkinson's disease. Only L-DOPA is given. It's also a plant compound. The L-DOPA is isolated from a plant called from the plant Atmagupta in Ayurveda. So in Tamil it's called Punekali. So from the seeds, this particular compound is isolated. So but that particular compound, when it is used, it has a lot of side effects. So what happened, when we combine the embolin and give this one L-DOPA in combination, it reduces the side effects to a great extent. So it's one of the important findings. We published a paper in Translation Neuroscience in 2032. And we studied the in silico binding studies also. Because I got trained, I'm very happy to mention here. I got trained by Dr. Divya Shanmugarajan, a senior application scientist of Altam Biotechnologies. She trained me in biotechnology. I attended a 1 week course in Chennai in Madras Veterinary College in August 2022. So I'm very happy to acknowledge your help. So with your help, I got some idea about this. After my retirement, I learned this subject and I started working with some people. And we found that in silico binding studies, the drug showed actually binds to alpha synuclein fibrils and break them. So that this alpha synuclein is a responsible thing for causing this PD. So we found that this one in the in silico studies. Now we are going for animal model, and later on if it clicks, we are planning to go for a patent and then clinical studies also. Okay, if time permits and people are interested to take up. Next one, please. Yeah. Now, not only that, we are now going for the derivatives or different forms of embalming. So the dimer of we synthesize the dimer of embalming, and then now we are focusing on the derivatives. Let's say the embalming derivative has been substituted, and particularly the experimental in the animal model, it gave increasing results. Increasing. Next one, please. Next slide. Yeah. Whenever we do good work, there are a lot of challenges. It's not, we cannot simply, people may think that, oh, take the plant, take the extract, ideally put the column, chromatography, oxygenate the column, and they do. It's not like that. Every stage, you have to be very careful. Okay, so even if you go wrong at one stage, everything will collapse. The whole system, procedure, the whole program will collapse. So right from the beginning, for example, collection and identification of the plant. We have to go to forests, we have to go to hills, and then collect the plant. You take the help of the taxonomies. So these things are there, issues are there. And identification, because many plants look alike. So you have to take the help of a pharmacokinetics, taxonomies to identify the purple plant. And the extraction, isolation, circulation take more time. So when I joined research, particularly in this field in the late 70s, so I was not that I was impatient. I wanted result very fast. So I had to work from the morning 7 o'clock up to night 10 o'clock. I was working. After 6 months, 7 months only, I used to get some results. So this type of thing, because I got used to it and developed patience. So like that, this type of research also helps us to change our lifestyles. And the in vitro in vivo screening. So to get the in vitro models, the cell lines, the bacterial strains, those things are not that easy. And there are a lot of issues. And in vivo so choose the right in vivo model, right animal model, that's also challenging. And once the particular active principle, the secondary metabolites, has been found out to be active, and it can be designated as a drug compound. For bulk production, you have to go for synthesis. It's not always possible to synthesize all the compounds, the enzymatic structure. Okay, then okay, we are done at one stage. Okay, we come to clinical trial. So you just undergo phase one, phase two, phase three, phase four to get the approval. Then approval procedures, you know very well in countries like India, it takes years. By the time you know drug comes, okay. I tell small, I explain these things with an example, then you will understand. Because these are not discouraging factors. Now more modern gadgets have come, computer systems, everything has come, biotechnology, bioinformatics. Now they are helping us to do these things to minimize the delay. So these things are. The next I'll tell you an example you can understand. Taxol. Next slide, please. You know, in countries like America, women get a lot of cancer. Most of the women are affected with breast cancer and ovarian cancer. So 1962, the plant was collected, Taxus brevifolia, and then first time 19, it took nearly 5 years to isolate the compound Taxol. I think the topic is heading is hiding. It's the Taxol, the isolation of Taxol. I'm telling you the chronology order. So in 71, only the structure was published in a journal. See, it took 9 years. Even in countries, in those days, of course, they had very minimum facilities. And you know, in 1978, it was found to have antimitotic activity. Another group, different groups worked on this particular compound taxol, very complicated structure. And in 1979, they published as a promoter of microtubule assembly. It means it's one of the important steps in the cancer progression. So once you inhibit that particular stage, so naturally the cancer will not grow further. Then 1984, this compound was subjected to clinical trial phase one. Then 1989 phase two. Then 1989, John Hopkins Hospital at John Hopkins University Medical College Hospital, they took us, studied, and then they published the paper on the activity in advanced ovarian cancer. The cancers are in the advanced stage are very difficult to cure. So remedy is not at all possible in majority of the cases. Whereas in the early stage, we can definitely have a breakthrough. So this type of particular taxol compound actually it worked in the advanced cancer stage, ovarian cancer also. So immediately in 1990, National Cancer Institute conducted an international conference on taxol exclusively for 3 days. It was a 5 days conference. So lots of people came and presented the papers. And in 1991, it was also found to have metastatic activity on metastatic breast cancer by the MD Anderson Research Institute in New York. It is also one of the very leading institutions. They are collaborating with the National Cancer Institute, and the large scale in Ausher pharmaceutical pharma they came up to produce this particular compound in large scale production. And the second National Cancer Workshop was done in 1993, and only in 93 the drug was approved for refractory ovarian cancer. So even in countries like America, where facilities were more, where the more importance given for research, it took nearly 12 years. Just imagine, it took nearly 12 years to make one secondary metabolite a drug. Okay, coming to the next one. Maybe the next slide, please. So these are some of the issues. So, and further, the environmental issue. As I told you in the beginning, is these secondary metabolites are present in very small amount, very minute amount. Okay, so what happened? So we would need more plant material. It means you have to kill more plant. Suppose if it is leaf or flower, we can, it's okay. Suppose if the activity is the roots, we have to kill the plant. So what happens? This leads to over exploitation of the medicinal flora and extinction of some many genera. So all over the world, people lost. So now what happened? The environmentalists they awoke and they became very cautious. They alerted the government to stop this kind of invasion. So in India, Botanical Service of India is maintaining a red data book on endangered species, and many plants which give many interesting good compounds are now listed there in the red data book. So without taking permission from BSI or from National bio agency, there's an agency by Government of India. We have to take the permission and then only test these plants. Okay, next one, please. As I told you, the drug, the taxol. As I explained to you, the drug of choice for breast and uterine cancers. So to treat one patient, at least 2 to 3 trees have to be cut to isolate the required amount of this compound. So it takes 12 to 15 years for a tree to grow. What happened? So naturally it becomes threatened species. Next one, please. So what happened? We have to go for alternate methods. So what are the alternate methods? Alternate sources to have the secondary metabolites. There are chemical synthesis. We can synthesize active compounds. Once the structure is known, there are synthetic analogs. What analogs? Just like having a body double in movie. The hero has to jump from fourth floor. So hero is a very costly person. he takes 100 crores for one movie, so he can't do it. So they employ a person looking like the hero. So they put makeup like that, like a hero. Then he will jump from fourth floor, of course, with some precautions. Okay, so synthetic analog. So what happens now? Similar to the structure, people started synthesizing many compounds, and then they started using. They become drugs. Okay, then biotechnological methods, large-scale culture plant cells, bioreactors for caustic products, and then for elucidation methods or metabolic engineering for improving anti-compound, I mean active compound. Like that, there are many methods. We'll see one by one. In another few slides, we'll be concluding our talk. Next one, please. Yeah. Synthesis of anti-compound. A simple example. Salix salva. It's called English willow tree. So people may be aware. Those who are cricket lovers, they may be knowing about willow tree. The cricket bat is made of the wood of willow tree, English willow. Its name is Salix alba. So people were taking the bark, taking decoction to get rid of pain, body, I mean, inflammation and fever in the olden days. What happened? The chemists came to the picture. They started isolating the compound, namely salicylic acid. Salicylic acid we can easily synthesize. So what happened? From salicylic acid, it was a responsible, secondary metabolite. So what people do now? There are a lot of restriction to cut the willow tree. So now from benzoic acid, we can easily synthesize salicylic acid, which is nothing but orthohydroxy acid. It's called salicylic acid. Then isolate it, we will get salicylic acid, which is called aspirin. In 60s and 70s, aspirin was in the market. But what happened? Aspirin started having side effects, gastrointestinal problems, ulceration of the stomach. So people stopped using. Now what happened here? New users come. In the past 2 or 3 decades, now more cardiac patients have come. People started affected by cardiac diseases, but particularly due to blood blocks, blockage in the blood vessel. So now they found out, they revisited the aspirin, they found out that it is thinning the blood. They removed the block. So what happened now? Now there's more demand for aspirin because on the lesser dosage, they're using an eco spray to treat the blood blockage. So now what for heart patients, this is a gift. Now the aspirin is giving gift. Now it is synthesized. No more, nobody is going to willow tree to this. This is a typical example of synthesis of the active compound. But origin is only from the plant, but it is now synthesized with the same property and all that. Phosphorylation gives better yield and better activity and commercially also viable. That's for another important factor. The commercial viability should be there. So even for 50 paisa or 1 rupee, we can get one tablet. Okay. Next one, please. So like that, many examples we can give. Then we go to the biotechnology. Biotechnology is of recent origin. The past 2 or 3, I mean 2 or 2.5, 2 decades only this field has come. Nothing but integration of biology and technology. See, once engineers never go to medical college, and doctors never see a engineering college. Now doctors are going to engineering college to teach biomedical engineering, and engineers are going to modern medical colleges to teach them engineering, other instrumentation, other technologies. Okay, the computer operation, those things. So now the fields become closer. Now there's no compartmentalization. So biotechnology is one among them. It holds promise in the field of plant research also. The tissue culture techniques are used nowadays to increase the yield of the active secondary metabolites, and they use biocatalysts in bringing about transformations and structural modifications. These are harmful chemicals. So nowadays, what happened? Your labs are now given warning by pollution control authorities. They should not use some acids, some kind of solvents like benzene. They should not use, and the concentrated sulfuric acid bulk they can't use. Like that, a lot of uses because the environment is getting affected. So now you have to go for green chemistry. It means you have to use the biocompatible materials, biocatalysts. So instead of going for harmful chemicals and which affect the environment, now biocatalysts are used now in the biotechnology to bring the transformations and structural modifications. Next slide, please. Maybe the next slide. Yeah, genetic engineering. This is another one important one. So production of transgenic plants. So what they do, when the plants grow, they change the genes so that the particular type of secondary metabolite they can harvest in large amount. See this particular example, Glycyrrhizia glabra. It's called Yashi Madhu in Ayurveda. It's used as one of the important neurological drugs. It's called one of the major rasayana in Ayurveda. In Tamil, it's called then Madhumegam, something for that they use it. So like that, digital is purpurea. These hilly plants in inside you can see the digoxin and digoxin are active principle. It's one of the cardiotonic materials. Now what happened, now they are being exploited in large amount by commercially oriented people. Now what happened, the genetics, they save the genes in the early stage itself, so that when they grow, the small amount of the plant gives more amount of the active compounds of the secondary metabolite. Then engineer resists against herbicides. For example, some plants grow very well. So when it is going to be harvested for secondary metabolite, some insect attack is there, some worm attack, or some kind of external factors. So now what happens? They develop some resistance using some genetic modification. So engineered plant lipids, like that some plant lipids from the plants or vegetable oils, they also now being produced in large amount by engineering the plants using the genetic engineering. So it's very important field. Lots of people are working, particularly CSAR labs like and ARI, Indian Agriculture Research Institute, ICR. Lots of people are using this method. Now the whole secondary metabolic production is slowly shifting to other people. Okay, that's what I discussed at the beginning itself. How the field is changing from and going from one end to other one for better. Next one, please. Make the next one. Yeah, fermentation process is one of the important factors in methods in biotechnology. For a typical example is production of thaumatin, a sweet protein. Diabetes people know diabetic people cannot take sugar or sweet items, whereas they need a substitute. So this particular thaumatin isolated of the African plant or from the African plant tomato Tomatopappus denali from the fruits, they isolated this particular compound. It's 5,000 times sweeter than sucrose, normal sugar, and it's a boon for diabetics. And scientists in the Unilever research lab in Holland, what they done, they isolated the gene for chromatin from this particular plant into the yeast cells. They isolate the particular gene which is responsible for production of this particular secondary metabolite. They cultured into the yeast cells. So what happened now? Now the chromatin is economically produced by fermentation process using genetically modified yeast. So now the yeast is producing, not the plant. Now nobody is touching the plant. So now commercially available in Japan under the name Tallinn. See how the field has changed. So now things are going in a different way. Of course, it needs lots of planning, organization, money, everything is there. The infrastructure and the role of experts, everything is there. Okay, next one. Next slide. Yeah, bioinformatics. We'll come to the final one. So the 3D molecular structures. What is the importance of bioinformatics? Informatics. It gives about the structures are given the 3D way. So you can see the structure from all angles. Normally, if you write on the board, you will get only two-dimensional figure. When you see the three-dimensional, we can see how the structure, where it is affected, which part of the compound, the particular secondary metabolite is attaching to the binding site of the disease molecule or by bacteria, virus, or something like that. So the binding sites are identified and possible activities are predicted using 3D. So sitting before the computer, you can predict if you know the structure of the compound. Suppose if you give the derivative, the informatics scientist, he immediately does all these things. Okay, it gives for isolation, scope for isolation of synthesis of new molecules by tailoring based on the predicted activity. So what happened, once you know this, so we know which type of compounds are present in which type of families of the plant. That all we can take. We need not disturb other plants. So it's also helping environmental issues also. Okay, to solve the environmental issues. Next one, please. So like that, every field is contributing to the development of this one. So I want to touch upon one of the important things, how the traditional medical system is useful. Can I take another 10 minutes, madam? 5 minutes? Hello? Madam
Yes, sir.
Simran, can I take 5 minutes extra? I have
Yes, sir.
another two
Yes, sir.
slides.
No problem. Yeah.
Okay. See, this one typical example where I worked, when I was working in the Ministry of Oil, we came across this kind of psoriasis is very serious disease. There's no cure for it. Many people are affected. I show the people affected by it. It's a chronic disease where the immune system becomes overactive and causes skin cells to multiply too quickly. So that you get fishy layer on the body. Just like fish, it has scales. Like the scales, you get the patient gets all over the body. It gives ugly look. So patches of skin become scaly and inflamed, most often the scalp, elbows, knees, and some other part of the body. Even I've seen patients all over the body also. So effective treatment aims to remove the scales and stop this skin cells to growing quickly. There's no complete remedy. Whereas in Siddha medicine, there is one plant called Rhytia tinctoria. From that plant, the scientists in our lab, we extracted the oil by Surya Kudan. In the process, we kept the oil extracted with the coconut oil. I'll tell the procedure later. Next slide, please. So from that oil, when that oil was applied, nearly 80% good results we got. See, this is the condition. This skin, just imagine on the skin, if this kind of scaly formation is there, how we feel. Okay, next one. Next one. I'm telling some 20 years old story. So this particular oil, we named it as a 777 oil, a coded one. So the leaves were soaked in coconut oil. They exposed to sunlight for 7 days. And on day 8, it was separated and taken and applied all over the body of the patient. And he was allowed to stand in the heat, sunlight in the morning and evening for 1 hour. We got 80% success rate. Only relapse only in 10% cases. Of course, along with some internal medicines also given, a total of 4,500 cases. It was a world record in those days. At the time, we were opening centers for Siddha medicine. And then after this, everybody started preparing the soil and selling in the market. So the formulation was slightly changed and patented and sold to a company. Now the company is, pharma company is producing this. It is giving very good result. Why I'm telling this? See, this kind of information is there with us already. See, modern medicine, there's no cure for it. But once identify the active compound, we can take to the modern medicine and use for this one, the treatment methodologies. Next one, please. Next slide. Yeah, formulations. All this for traditional using formulations, to enhance therapeutic activity, plants are collected, they have some specifications. Plants should be collected only during specific times because at the time only the secondary metabolites are more there. And similar activities are combined, plants with similar activity. The ingredients are processed, they are not used just like that. Okay, they are not used as such. The final products have specific indications based on the durations, dosages, and the vehicles. These things are very important. So preparations like chornam, as I told you, they are in the already market. They are used in large numbers. Next one, please. Why I'm showing all these things? Things need more attention of scientists to going deep into it and doing more good things for the people. Next one. I think last one. Maybe the next slide, madam. Yeah. To conclude, so what have I told? For first 1 hour, last but the penultimate slide. Plant research is a broad scope for all specializations. The future life of people depend on plants and plant-based products. As I told you, people are going for environmental friendly products, environmental friendly homes, environmental friendly clothing, everything now. So plants are play vital role. Besides food, clothing, shelter, and medicine, nowadays plant products find as useful fuels. Government of India is going to have 20% ethanol in petrol. So I think policy has already been made by central government of India, to reduce the pollution. Okay, they are safe and environmental friendly. As drugs, they are safe, effective one, exact least side effects. And India is an ideal place because we have for this kind of research because we have very suitable ecological, geographic, and agroclimatic conditions. So we have to make use of it. Okay, next slide, please. I want to acknowledge this Alchem Technology Bangalore for giving me this opportunity to share my experience with the young minds and particularly my mentor, Dr. Divya Shanmugarajan, Senior Application Scientist in Alchem Technologies. She actually gave very good information about bioinformatics, so I learned a lot from her. I'm grateful to her. And of course, the coordinator of this program, Ms. Simran, as Ms. Giti, for arranging this event very nicely. Thank you all. If you have any questions, you can post or you can ask. Thank you very much for your patient listening. Next slide.
So, thank you so much, sir. This was a very knowledgeable session, and it's time to take some questions now.
Yeah,
So,
please.
if you have any
I'm all.
questions, please post them. So, our first question is, which drug
Yeah.
delivery method can be used for indoles so that the bioavailability can be increased for it to inhibit estrogen related cancers?
See, actually, thank you very much for this question, Ms. Pooja. But I'm not a pharmaceutics person. No, we are not. My area is not drug delivery. Okay, We are only just giving the idea how this plant from the plants, the drug compounds can be isolated and put to use. So how in modification, in what form it should be, either it should be in injection form or capsule form or tablet form.
Okay.
That those things have to be decided by the pharmaceutical people. So they will find out, or because it varies from compound to compound. So the method what we
Okay,
use
sir.
for alkaloid cannot be used for indoor. Okay, there are many methods to increase the bioavailability, particularly for as I told for estrogen and related cancer type of cancer. There are many methods. So I'm not expert in that. Sorry.
All right, sir. Thank you.
Any other question?
The next the next
Yeah.
question is, what's the difference between Ayurveda, Siddha, and Unani, and which one is the most effective in treating a disease?
As I told you in the beginning itself, Ayurveda developed from North, originated from North India, and its literature is in Sanskrit language. Okay, only people who know Sanskrit can read it, the original text. Now translate books are available in English, other languages also. Like that Siddha, it originated from southern part of India, and its literature is in Tamil. So unless you know Tamil, we cannot read Siddha literature. Even people who know Tamil, for them also difficult because they use grammatical Tamil, the old Indian Tamil. Because Tamil is one of the classical languages like Sanskrit. It has undergone a lot of changes. So the language we talk now is entirely different from what people at the time spoke. So it's in on that language. So there are some people who can interpret those things. And Unani, as I told you, the literature is in Urdu. So people who know Urdu only can read the original text. Okay, this is the basic difference between these things. And all these things, the common factor is all these things use plants, metals, minerals, and animal products as drugs and formulations. Okay, as drugs. So as I told you, these traditional medicines mostly they are very effective as prophylactics to prevent or to take care of or to improve the immune system only the health. As a curative, they are not that effective, particularly in the advanced stage. See, for example, modern medicine, they can go up to the modern medicine. Say, for example, cancer. Different stages of cancers are there. Early stage, middle stage, advanced stage are there. So early stage, they can treat using conventional medicine. In the middle stage, they use some radiotherapy, chemotherapy, and other things, and surgery also they be involved and other things. Advanced stage, very difficult. So they leave the patient to die because it is gone that level. So there are restrictions. So no, we cannot compare between system. This is good, this is better, and this will give. No, it depends on the disease, depends on the age of the person, it depends on the comorbidity. Take for example COVID-19. What happened? COVID-19. What happened? The people who succumbed, they had problems. Most of them were drug addicts, smokers, alcohol consumers. Then they had comorbid disease or problem or diabetics. Only they only succumbed. People who did not have these things, they survived. Is it not? And different age factors also there because as we age, the immune system goes down. Okay, so naturally that's why people get age. When we get age, they get disease. In Tamil we say there are four stages between life and death: graying of hair, okay, then forming what we can call cataract formation, okay, then disease, and then losing, faculties going down. In Tamil, we say different way. Nari, Thirai, Pini, Mupu. We can tell. Nari means graying of hair. We can't stop it. Once you cross 40, 50, naturally I'm telling you, so you don't use shampoo and other hair colors and everything in the young age. Nowadays people are getting gray hair even at 20 years. Okay. Normally, normal person, a healthy person, in those days I'm telling, some 50 years back, 60 years back, I have seen, my father got gray hair only after 60 years. Okay, like that. So graying of hair. Then Thirai means cataract formation. See, it is automatic aging process. Because eye vision, goes down. So because of so, you have to go for remove the cataract and then wear glasses. Okay, Thirai. Then Pini means disease. So automatically, because our immune system goes down as we age. It's common for all living organisms, including humans. Okay. Then Mupu means faculty is going down. That's why aged people cannot hear. If you have to talk louder to them, you have to go near and shout. Then only they can hear. They can't see distant objects. They can't feel. Giving some ants bite, they won't feel because the feeling sense, everything sensitivity goes down. So these four factors are there. So this is for this also there are many stages. See, for example, cancer in young age, that's cured easily because body resistance is there. Okay, different drugs can play vital role, but in advanced age they can't give. Even the middle age surgeries are done, effective. So medical field is very big field. So these kind of questions, normally people get. Even I had these questions when I entered the field in the research. I used to discuss it. I had a lot of friends in these things, so I asked these questions and gathered the information. Okay, that's why I'm sharing with these people. It's a very good question. Because many people ask. See, supposing someone fine morning they get a call. They say, my relative's got cancer. He went to nearly 10 doctors. Now everything is okay, over. They told no cure. Can your system cure? Can you help me in identifying with the doctor to cure? Then we have to go through papers. I recommend to some of my friends, or I myself go and help them, and then tell the position. See, this is the advanced stage. There's formed metastasis. Second stage has come. So now it's very difficult. Your age is not cooperating for surgery. So these are there. So point blank, we cannot tell which is good. Difference we can say. What are the differences between this medicine? The treatment methodologies vary because they originated from different places. Say, for example, in Ayurveda, plants are more because it originates from North India. The Himalayan region, whole region is there. Whereas in Siddha, plants are very less compared to Ayurveda, less plant of plants, whereas more of metals and minerals. So Siddha used more of metals. So recently I edited one book to CRC Press of Taylor Francis on zinc. So we have 315 pages book, that one. Exclusively on zinc. So in that I have written about use of zinc in different traditional medicines. So like that, copper, gold. So gold is used extensively in Ayurveda. Tanga Baswam. The former Chief Minister of Tamil Nadu, Dr. M.G. Ramchandran, MGR, cine actor. He was using more of this gold basma. And his body used to be gold in color. I have seen him very close. Because he was a bodybuilder, and for just a health tonic, he has taken that one as a health tonic. Of course, with the prescription by the doctor, Ayurvedic doctor. Like that, there are lots of therapeutic things in this system. You have to identify which one is good, which one is bad, depending upon a factor. So, say, for example, in China, what happens once a patient enters the hospital, all specialists are there. So they are screened first by Chinese specialists, Chinese medicine specialists. He recommends, he discusses the patient tells, see, this can be cured by Chinese traditional medicine. He takes them. Something he will tell, no, you go to modern medicine, get diagnosed on these things, then you come back, we will see. Some of the things he himself says, no, you go to modern medicine, our system cannot cure. So this is called integrated system of medicine. Now government of India is planning to have this integrated system of medicine to integrate all. So for that, the mindset is very important. See, the modern medical doctor should accept the traditional medicine. The traditional medicine doctor should accept the modern medicine. So then the integration can come. Then we can say. So for my, see, I have 4 decades of experience in this field. What I have seen, integrated medicine will be better for diagnosis and elucidation of the disease. Prognosis, we can use modern medicine. For treatment, we can use these traditional drugs as a supplement, as adjuvant. That is possible. Okay, that is possible. Okay, for example, for a cancer patient, if they give radiation therapy, hair falls. So in that case, you can use some plant medicines to control hair fall. Okay, those things can be done. Okay, then their red cells go down very fast when they give surgery in the cancer. So in that case, they can take some traditional medicines or plant drugs to control to increase the blood cells. I mean red blood cells. Like that, we can use as adjuvant. That's my way. And I talked about these things in meetings also. If you arrange, I can give this presentation also, no problem. Okay, thank you very much for this question. It's a very good question. Next one.
Next question is how to standardize the concentrations used in a formulation that has various compounds.
Yeah, this is a challenging question. Now in the rounds in government as well as in pharmaceutical companies, everything. See, there are different methods of standardizations. Okay, what happens unfortunately uh, in the institutions where I work and even the government institutions where they are going for standardization, they adopt the modern medical parameters. Mostly it won't work. This is my personal view, I'm telling. Of course, I talk this in the committee meetings also, even whenever I chair sessions or I go for lectures and all that. You cannot use the same thing. See, I tell small example. I always give the one same example. See, take for a jet plane and bullock cart. Both are used for transportation, right? Both are used for transportation, is it not? If you go to a village, you have to go only in bullock cart. We have no proper roads and all these things. Only those things are available locally. You have to travel. If you want to go from one continent to other continent, you have to go only by jet plane. Both are used for transport. So we cannot interchange. I can't say I want to go to the cut and go from Australia to America. That's not possible. Is it that? Same way, others, I can say I want to take jet plane to go to my next village. We cannot because these two things are different. Same way, see, each system has its own way. As I told you, the standardization, we worked on many plants. What we found, the methods told in the literature itself. Say, for example, if you are taking one particular plant, see, Bhumi amala it's called, which is used for jaundice. Okay, in particular season only, the active compounds are more in that one, particularly the rainy season. Of course, the plant itself is available only in the winter. Only if it rains, it is very effective. During summer, it won't come, but some places it may be there. But it's not that effective. That's what we found out, okay, in our research. Okay, so that's why we have to collect during that time and use it. It will be effective. That's the standardization. Okay, that's the standard for using Bhumi Amalaki, okay, or Bhumi Amla. That is the method. We can use modern parameters if it is compatible. So we cannot incompatible things we cannot do it. Okay. Say for example, the viscosity of a particular drug. See some drugs are in the form of viscous liquid. Okay, viscous liquid. Then we can use viscosity as one of the parameters. So that's what we recommended. Even I was in some committees, I recommended some of the parameters which are useful to standardize. Okay, some of the parameters which we cannot use, we should not use it. Okay, are you okay with my answer, madam? Who is this? Ah, infantry. Who is? Yeah.
हाँ चलो वो तो वक्त ही उसमें होगा सिविल वक्त को डाल देगा
Do we
Okay,
have
any
any more
question?
questions?
कोई got answer
Hello, sir. What are the factors
Yeah.
affecting plasma
कोई
protein
अच्छा
binding of
skill
small
है वो
molecules?
What are the factors affecting? No, madam, I am not expert in this. I am not expert in this field. Okay, this you have to ask
All
only
right.
the biochemist. Okay.
No problem, sir.
ये है ना thingiverse
Any more questions?
चलो
I think I've taken more time.
जैसे
I think that's it, sir. I don't see any more questions.
Okay, no problem.
कुछ estimate चुनो जो तुम्हें print करना है सारे simple SQL है ठीक है ये भी simple है ठीक है
So, thank
और
you so
कुछ
much,
ऐसा हो
sir,
जैसे ये सब
for
कुछ
this
ऐसा
very
हो जो
knowledgeable session. And
उनके
it's
लिए
now time to
मीनिंगफुल हो
felicitate our
जनरली
speaker
मीनिंगफुल
of
में
the day with
लोग
the
एक्साइटेड हो
certificate
जाते हैं
of
मोबाइल के स्क्रीन
appreciation.
देख के कोई
So,
छोटा मोटा
thank you so
स्टेटस
much
देख
once
के
again, sir.
ठीक है
Thank you, madam.
एक्चुअली
Thank you very much.
ये क्या है
And also, I'd like to thank all
स्टेप
of the
तो
participants for
प्लेसमेंट
being
तो
part of this
ये
session.
होगा नीचे का
So,
और
please
देना
stay tuned with us, everyone. Please leave a like, and to get updates for more informative sessions like this, please subscribe to our channel and follow us for more upcoming events. So, thank you so much.